And thank you very much, General Counsel, Deputy, and the Physikalische Verein for hosting this Panta-scientific evening. I hope all of you will postpone your lethargy, but in any case, we will for sure not be allowed to go in that state by the fantastic words of my dear friend and colleague, Walter Tinganelli, which I'm very honored to present this evening. So Walter is from Napoli, where he got his master in biotechnology at University Federico II. And then he started the typical life of a scientist, moving around, first year coming to Germany, to nearby University of Darmstadt, where he got his PhD, and then traveling a bit around the world, in Japan, back in Italy, in Trento, where he started various activities, among the most important, probably the setting up the first laboratory for experimental room in proton therapy in Trento. Now back to Germany in 2019, he's actually group leader at the clinical radiobiology group at GSI in Darmstadt, where he conducts, among others, that he would like to discuss also with you at some point, but this very fantascientific topic of hibernation and how this can help in many aspects of life. Walter, thank you very much.
Thank you very much. Thank you for the very nice introduction. Good evening, everyone. It's an honor for me to be here tonight to speak about synthetic hibernation for space exploration and medical advancement. But my final goal is to show you that synthetic hibernation is not science fiction, but it's something that can be more real. I do have other projects at GSI in the clinical radiobiology group. As the name says, clinical radiobiology, we do a lot of things about, we study the metastasis, we study new way to do radiotherapy and so on. But the synthetic hibernation is the most fascinating one because it's on the edge of what is the science fiction. And that's why I like to speak about this specific topic. So just as introduction, space is an hostile environment. We know that. Astronauts in space, they are in confined space in which they have change of mood, nutrition, fitness, they have sleep disruption, they have like completely disrupted circadian rhythm and so on. They are isolated, they are far away from home, even though they are actually just 400 kilometers from the surface of Earth in the International Space Station. And they are subjected also to other, let's say, problem in space. One of these is the shift of the fluid, as you see here. So from the bottom side, the fluid moved to the upper side. And then most of the astronauts, the astronauts have this puffing phase. This puffing phase, because of the fluid, also produce like a small pressure on the eye. And sometimes the astronauts, even if they have like a very good view on the Earth, they need glasses in space. And then, of course, they are exposed to two of the major showstopper that are the microgravity and the radiation. Above microgravity, astronauts in space, they have bone loss, muscles atrophy, they have oxidative stress, and for radiation, but we will see in detail, they have risk of cancer, cognitive decline, visual, as we said, cardiac, and so on. But we will see in a second. So natural radiation is everywhere. On the Earth, we are exposed to radiation all the time by radioactive soil and rocks. We are exposed to the radon, that is a gas that is produced by the ground, and we breathe, especially people from the Naples that live in a volcanic area. There is a lot of radon in that case. Our bodies, even our bodies are emitting radiation. So right now, the person close to you is radiating you. So just to say. And then, of course, there are cosmic rays, but we will see in detail about the cosmic rays, about the space radiation, and even plants and fruit. So even when we eat plants and fruits, we are exposed to radiation. And actually, there is what is called the banana equivalent dose. It's a little bit stroboscopic, but this was not my intention. I don't know. Okay, thank you. I just continue. In any case, I mean, for the banana equivalent dose, in principle, if you eat like 50 banana, you have the same radiation that you get for a dental X-ray. If you eat like 400 banana, is the equivalent dose that you have flying from London to New York. And if you eat 100 million banana and you survive for a couple of weeks, you have a dose of radiation that actually is fatal in a couple of weeks. You will have a lot of other problems, I guess, so don't do it at home, yeah? But in case you have like the fatal dose because of that. And then the space radiation, I guess, was the banana equivalent dose that was like the strange slide. For the space radiation, essentially, what is the space radiation? Essentially, space radiation is are atoms without electrons, so are ions that are accelerated very fast. So they are a little bit like very fast bullets. And this space radiation, in principle, come from galactic radiation, galactic cosmic rays, so from faraway galaxies, supernova, and so on, but also from the sun. The difference between the two is that the galactic cosmic rays, they are very fast, very energetic. And then they can go through essentially any kind of material. And in any way, in space, to bring material, it costs a lot of money. So then essentially it's very difficult, if not impossible, to really protect efficiently from the galactic cosmic rays. The solar radiation, the particle emitted from the sun instead, are still particles, but are less energetic, and then still is possible to protect the astronaut from that kind of radiation. But then the sun sometimes produce what is called the solar particle events, when there is like a huge flux of particle that is released from the sun. Astronaut in this case can repair inside the particular region of the International Space Station, but if they are in an EVA, extravehicular activity, this can be lethal for them. And if it's not lethal for them, actually something very nice happened to us. This is a picture that actually my niece did a couple of weeks ago. She lives in Iceland. She's lucky at least for this, because for the rest, Iceland, it's tough. But then you see the particle, they arrive to the magnetic field of the Earth, because the Earth protects ourselves from the radiation with the magnetic field and with the atmosphere. So when the particle reach the magnetosphere is deflected, then this particle impact to the atmosphere, and then you have the wonderful northern light. So we see this wonderful coral, but actually these are particles that would like to kill us. But instead we enjoy and appreciate this wonderful color in space. Then galactic cosmic rays are isotropic, so essentially astronauts are, there is a new one, let's try the new one. Perhaps this is, thank you, I can take it. Thank you. Sorry, but perhaps this works better. In the end, we have 1 hour and a half. So let's see if, so now there is nothing at all. Okay, now wait, now should work. Yeah, let's see if this works. Okay, so astronauts then in space are essentially bombarded by the galactic cosmic rays from 360 degrees. So let's say when they are close to the Earth, of course, they are protected a bit from the Earth, but then in space, they are 360 degrees exposed by the radiation. And actually, this kind of radiation is 100, 300 times higher than the radiation that we have on the Earth. So on the Earth, the average dose of radiation that we get, now we just see some number, is 3 millisievert per year. The astronaut in space, they get around 1 millisievert per day. So it's like a very high dose of radiation. But in any case, when you, just a curiosity, when you speak with the astronaut and you say that, you know, in space there are radiation, you will be in danger, they don't care. Because essentially say, I'm traveling in a rocket, I'm going in space where there is the void and so on. I don't care about radiation. But the first thing that they ask when they come back is, okay, how much is the dose that I took now? What happened to me now? Because after they are scared, but the beginning, they are scared for other reasons. Moreover, even the sun has a magnetic field. And then the magnetic field of the sun is also protecting ourselves from the galactic cosmic rays. And actually, the sun is a magnetic field that has a cycle of 11 years. And actually, if we want to do mission deep in space, like on Mars, or I don't know, in some satellite of Jupiter and Saturn and so on, we should most probably consider to travel during the maximum activity of the sun. That is like a counter-interactive. But the point is that when there is like the maximum activity of the sun, we are protecting the galactic cosmic rays. And remember, the galactic cosmic rays are the most dangerous because they are very fast. And then, yeah, I mean, this is also now what the Mars Express found, that essentially the solar activity increases, then you have fewer cosmic rays. And then essentially until now, with the International Space Mission, we were in the very low Earth orbit. So we are something like 400 kilometers from the Earth. But now we are moving a little bit far. And now you see, most probably you heard about the Polaris Dawn mission, where it was the first human mission to travel so far from the Earth since the Apollo mission. And they also had an IFA in this case, where you can see down here, or perhaps you cannot see because it's too small, but they did an IFA at 736 kilometers from the Earth. So this was like very far. And before this, the only person that actually traveled far away from the Earth were the men from the Apollo mission. So these actually are the men from the Apollo 1, White, Sheffy, and Grissom, that unfortunately they die for a fire accident. But after them, men from Apollo 8 and 10, they travel around the moon. And then from Apollo 11 to Apollo 17, not included the Apollo 13, that is very famous also from the movie from Tom Hanks, they were able to land on the moon. And then we have 27 humans around the moon. We are speaking about 380,000 kilometers average from the Earth. And just 12 humans walked on the moon. But now the plan, as we said, is to go back on the moon. So you heard most probably about the Artemis mission, but also move on Mars. So the idea is to have like a new space station around the moon now. So to do not use anymore the International Space Station, but to have another one around the moon, Gateway, and actually to have like moon colonies. And after that, the plan is to go on Mars. But we see that the farther we move from the sun, the more increase the average dose of radiation. So if we go to Mars, then we have to consider not almost 1 millisievert per day, but we have to consider also almost 2 millisievert per day. Remember that the average on Earth is just 3 millisievert per year. There are also cases of people that live in place where the average dose is a lot higher. For example, there is a place in Iran, Ramsar, where they have actually a place where the dose of radiation is 130 millisievert per year. And the point is, in this place, actually, people have a less percentage of cancer. Now the question is, they have less cancer because the radiation somehow stimulates the immune system and they have like a better immune efficiency that can kill the cancer cells and so on, or they are selected because of the radiation and then you have just a very radio-resistant person living there. Nobody knows, but this is one question that in principle scientists are asking. And then you see this is now a nice data that we get from the Voyager 2 that is a probe that was sent from the Earth many years ago and now actually went over the heliopause. And actually over the heliopause, you can see that there is like an increase of galactic cosmic rays and a drop of particles from the sun. So it's really like the sun is protecting us. And if you go over this shell, then you are bombarded by this very dangerous radiation. And what the radiation does to the cells? These are, this is one picture and one GIF actually of a nucleus of a cells, in both cases, traversed by iron. Because in principle in space, you remember we say we have like bullets, we have particles or atoms without electrons, and actually these particles, most of them are protons, so very small bullets. But then we go up to ions, and then the ions are very heavy, and when they go through the DNA, so you see the nucleus of a cell with this gray color, and then you see two tracks of iron going through, and what you see, this fluorescence that you see, actually is the damage that is produced to the DNA. So at this point, the cells can do three things. One, say, okay, there is too much damage, I'm going to die. It's called apoptosis. It's like a programmed cell death. So they say, too much damage, I have to die, and the cells die. The cells can repair the damage efficiently because we have a lot of molecules for the DNA repair. But in this case, where there are a lot of DNA double-strand breaks, it's very difficult that the cells is able to repair the damage efficiently. And if it repairs the damage in a not efficient way, you can have mutation. And then mutation can bring, at some point, to cancer. So traveling to Mars, in a transit to Mars, we have to consider that every cell will be traversed by one proton every 3 days, every cells, by one helium every 30 days, and most of the cells will be traversed by iron. So, and what actually radiation then can do to a man? So if you are lucky, something like this can happen to you. A little bit less lucky if you become the thing. But yeah, if you are lucky, this can happen to you. If you remember the movie, essentially they were in an IFA and they were exposed to solar particle events. And then they come back on the Earth and they had like wonderful power. This actually is difficult to happen. Yeah, I mean, we are studying if something like this happens, but most of the time you can have cancer, tissue degenerative effect, like in the central nervous system, cardiovascular, cataract, or again, you can have the acute syndrome, but this is for the solar particle events. But then when we look at the risk, so how much will be the risk of cancer in the astronaut? You see, for the different mission, International Space Station, Lunar Colony, Mars mission, there is an increased percentage risk of cancer death. And you see also for Mars, you go between 1 and almost 13. There is a huge variability. But why we have so much variability? The point is here, radiation quality and dose rate effect. Not many human beings have flown in space, so we don't have data for this. Most experiments conducted in ground-based facility. GSI, for example, is a ground-based facility from the European Space Agency. So if you are a scientist and you apply for the European Space Agency, you want to do an experiment on a plant, on an organism, cells, whatever anything you want to irradiate, you can come at GSI and we can irradiate with the iron, with helium, with everything you want. The point is, in space, you are exposed to radiation, the dose rate is very low, and at GSI it will be a little bit too much expensive, so we cannot do this for you. So we can give like we can give 500 millisievert, 1 millisievert, and so on, but in a very short time. And this is not really what you have in space. Also, the radiation quality. Most of the data that are taken for that kind of graph, they come also from patients that were treated with radiation therapy. But the patients treated for radiation therapy are treated with different of radiation, not with the iron that is very heavy. And then when you change the particle, when you change the type of radiation, you change also the effect that you have in the cells. For the tissue degenerative effect instead, as we said, this can be central nervous system, cardiovascular, and cataracts. And then there are experiments. So astronauts know that it's quite common that when they become aged, they can have cataract. But cataract is not a big problem. The problem would be like the cognitive decline. Imagine you do like a long trip on Mars, you land on Mars and say, okay, what I'm doing here, what I have to do. So this is actually not the best scenario for an astronaut. And then there are experiments done with animals. So here you can see again, this is like an acute dose, so high dose rate. So you give all the dose in a short time, it's not chronic and so on, with oxygen and titanium that are quite heavy in mice. And there are two different tests, novel object recognition, object in place. So on the x-axis, you have different ions, you have different doses. On the y-axis, you have a discrimination index. And then you see that increasing the dose, you have like less ability of the animal to recognize a new object. So the mice are very curious. If you put an object inside the cage, the animal want to explore, want to spend time close to this object. If this time starts to decrease, it means that there is a cognitive decline to the animal. And then you see there is a clear effect, but again, with an acute dose and so on. Then there was a nice experiment done actually with just two subjects. So this is not the best, but two twins. So one was in space while the other one was on the ground. And then what they did a lot of study, biochemical, cognition, epigenomics, gene expression, immune, etc., etc. And they did test pre-flight, in-flight, post-flight, just to see what happened. Then in the end, they collect all the data, and it comes out that microgravity, as we knew, microgravity and radiation are the two major showstoppers. Microgravity produces body mass changes. So again, in space, since you have no gravity, you have like the loss of your muscle strength, you have loss of calcium. 2% of calcium from your bone is lost every month in space. Then they have to do 2 5 hours of exercise per day, the astronauts. So I wanted to be astronaut. This was the point why I did not. No, actually, they didn't take me, let's be honest. And for ionizing radiation, there is like telomere length regulation. This is not important right now. Chromosome stability, cognitive performance. And then you see predicted risk for 1 year Mars travel. The biggest problem are tumor development and cognitive decline. So the astronaut can develop a tumor, even if not during the mission, but after, and can have a cognitive decline. And this again can be a problem if you are traveling in space. That's why there are limits for the astronaut in space. So you see different space agencies, they have different limits. So ESA, Russia, and Canada, they use like 1 sievert. This is the career effective dose. So in your own life as astronaut, you can take this dose. But if you go to NASA and JAXA, then there are differences. You see, there are differences between the age and between the gender. For the age, it's clear why. Because if you are older, even if you take radiation, you have less time to develop a cancer. But for the gender instead, the point is that female have organs that are more at risk of radiation, like breast and ovary. The point is now they try to find a way to have only one limit for everybody, because imagine the situation now. We have a mission to Mars, and then there are people from NASA, people from ESA, people from Japan. They start different gender. They arrive immediately after the moon and say, sorry, guys, I have to go back because I go over my limits right now. So they are studying now how to have one limit for everybody. And how can we protect astronauts in space? So the possible shielding, again, galactic cosmic rays, they can lead even over 100 centimeters of any material. They are very fast. It's very difficult to stop them. On Mars and on the moon, actually there are plans to use 3D printer robot that actually will take the regolith. Regolith is like the sands that is on the moon and Mars, and to print structure that will give some protection to the astronaut. Here, you can exaggerate with, with the material that you put, because you have a lot of material. You do not have, let's say, excessive cost to send something in space. But in space, we have a problem because we have to reach the moon. Actually, on the moon and on Mars, they are also searching for natural cave, because the natural cave will be the right place where to put the astronaut. However, on the moon and Mars, there are other problems. For example, on moon, There is no atmosphere, so you have like radiation is bombarded constantly, this regolith, this sense. And then this actually is very sticky and very radioactive. And for example, the astronaut from the Apollo from 11 to 17, they had this kind of problem because when they enter in the LEM, so the Lunar Excursion Module, they bring with them a lot of this sense. And if you breathe a source of a radioactive source, is even worse, of course, than having from outside. There are also ideas to use active shielding, but these at the moment are not really feasible. So active shielding essentially is to imitate the sun and the earth. So to build with some big magnets a magnetic field around the space station that can deflect the particles, but these are very difficult. And then there is another way. Well, of course, there are plans to give astronauts food with antioxidant, drugs. There are a lot of things, of course, but now there is also the idea to use hibernation. And now we enter into science fiction. I know most of you think that now we enter fiction, but just to clarify, hibernation is not cryoconservation. When we speak about hibernation, we don't want to bring astronauts in minus 200 degrees in nitrogen and so on. No. These actually, there are companies that do this, like the Alcor that is in Arizona. I don't remember now the price because I'm not interested, but you can pay like 100,000 euro and they come to you, wait that you are dead, so they stay close to your bed. When you are dead, they substitute your blood with cryoprotectants. They take your body, they put in liquid nitrogen, and then you stay in liquid nitrogen until technology is good enough to cure your cancer or the things that kills you, and until they are able to wake you up, because at the moment there is no way to wake up a man from minus 200 degrees. So this is a different story. We are not speaking about cryoconservation. We are speaking about hibernation, that is, with my horrible German, Winterschlaf, letargo in Italian. So where essentially you have like a drop of the metabolism, you have a drop of the heart rate, and you have a drop of the body temperature. But how much we can decrease the metabolism? You see here, small animals, they have like a very fast metabolism because they are small. To keep the worm, they need to have a very fast metabolism. Big animals like elephants or whales, they have a very low metabolism. So it's very difficult to bring an elephant in hibernation because his metabolism is already very low. But a human, there is still a small margin to decrease this metabolism and to bring this person in hibernation. And then there are different animals, the bear, for example. So you see, in principle, in the hibernation, you have like facultative hibernators and obligate hibernators. The facultative hibernator are those animals that when they have scarcity of food, when it's very cold, but we will see hot, they decide to switch off their metabolism and to go in hibernation. And then there are the obligate hibernator. The obligate hibernator, like some squirrel, Arctic squirrel and so on, if you take the squirrel in a very nice place with a nice temperature, with light, with food and so on, he will continue to go in hibernation at the same time every year and so on. These are obligate hibernators. But all of them, they have more or less the same situation. After the hibernation, they are active. They go in hyperphagia, so they start to eat a lot. I like this because I can say that I'm not fat, but I'm hyperphagic. So I'm preparing myself for the hibernation at some point in life. And then they enter in hibernation. And then you see here, the bear in April and in October. Actually, at the beginning, there was also an idea to do not consider what the bear do hibernation, because you see, the body temperature of the bear, it goes from around 37 degrees to 34, 35, not more than this, just a few degrees. But he switch off the metabolism, lose the weight. Why it lose the weight? Because he needs the fat, the protein, and so on. For example, he just recycles his own urine. and he used the urine just to protect the muscles. And he also used what is called the water, metabolic water. So 100 grams of fat is able to produce around 107 grams of water. So essentially use this kind of water. But during hibernation, actually bear can give birth, so can have like almost a regular life. And you see the body weight then decrease, then increase a lot because they start to eat hyperphagia, and then they enter in hibernation. The ground squirrel instead, now you can see here, active season, and then it starts the hibernation. We will see in a second why we have all this peak. But you see the temperature is going down, down. It can go even lower than 0 degree. And the question is, how can it go to 0 degree without freezing? And that the blood of the squirrel during hibernation, the blood, you have no platelet anymore. All the immune cells, they migrate to the lymph nodes, so the blood is completely free from any particles. And in this state, it can become a supercooled organism. So essentially, since there are no particles inside, this bottle is already down to 0 degree, but there are no particles inside, it's completely clean. Then you have no ice. But then when you touch the bottle like this, you produce some impurity, some gas inside, and then you have the ice that is forming. So the animal in this case, they do not freeze because they are super cooled. And then you have something like the tail dwarf lemur, that actually this was the first primate that was found able to go in hibernation, even if it's not hibernation, but this is called an estivation. So what is estivation? Essentially, as I said before, is not just about cold food and so on. In this case, when it's too warm, this animal, and there is no water and so on, this animal, instead of dying, just decide to enter inside a tree, switch off the metabolism, and when the animal switch off the metabolism, doesn't mean that the temperature drops. It can be that if outside the temperature is higher, actually the body temperature increases. So then they are in hibernation, but it's called a estivation because the body temperature actually can increase in this case. And then, as we said, hibernating bears, they conserve muscle strength more than human on bed rest. So again, astronauts in space, they need to do like 2.5 hours of exercise per day because otherwise they have problem with their muscle strength and with the bone. And actually this study is done comparing, let's say, the bear with the human on bed rest. So what is bed rest? It's a kind of experiment that you can do actually, even ESA, they pay, I don't remember now how much, but they pay you something like 2,000, of course you have to be selected, but they pay like 2,000 euro per month just to stay on the bed. But believe me, it's not really nice. I did not, but I tested because I wanted to see, and it's horrible. So to stay like this, and then, I mean, someone is washing you, someone is giving you food and so on, but they do experiments like this to simulate the microgravity, but they pay a lot of money. So if you are interested, you can still apply. But then what happened to the bear? So the bear are able to protect themselves, but how they do that? So there is a nice experiment that was done, taking the blood of the bear during the summer season and during the winter season. And you will see that this is not the first time that they take something from an hibernators and they use it for another animal, or in this case, for human cells. So they take muscle cells from a man, they cultivate inside, these are the normal flasks that we use in the lab for cultivating cells. So what you see in pink actually is a medium, so it's like nutrient for the cells. The cells are attached to the bottom of this flask. And then usually we have like a general medium that contains some amino acid and so on, and then we put inside the fetal bovine serum that is like nutrient for cells. In this case, they did it with fetal bovine serum, but they also tried the summer bear serum and the winter bear serum. And then what happened? In principle, it happened this. You see, up left, you see the fiber, the muscle fiber with the fetal bovine serum summer bear serum, winter bear serum. Winter bear serum, when you put the winter bear serum inside the flask with human cells, human muscle cells, you have like bigger fibers. And then you have the degradation rate of the protein in these muscles decrease with the winter bear serum, and also the protein degradation decrease when you put this winter serum inside human cell line. So then why to hibernate? Because we want to survive to temperature, we want to survive to scarcity of food, water, predators. There are different kind of hibernation. We saw it. So essentially, the endotherm animal are the animals that are able to produce their own energy. They keep the energy constant. Homothermic, they are also able to produce the same kind of energy. Poikilothermic are those animals that are able to change the temperature during the time. And ectotherm are actually those animals, like for example, the snakes, that they are cold blood animals, but then they need the sun to become warm. And then in this case, we do not say, it's everything more or less the same. I mean, the mechanism is the same, but in this case, we call it brumation, because it's not them that regulate their own metabolism, but it's the sun that warm them up. And the hibernation, we went back to, you remember, we said this different peak that we saw before. We go back on this. The hibernation is not a stable process. It's not that you enter in hibernation, you stay in hibernation. You enter in hibernation, there is a drop of the temperature, and then you have these arousals. Why this happens? This is still a mystery, but there are two hypotheses. The first hypothesis, animals during hibernation, they are not sleeping. They are in a non-conscious state that it's really something close to the death. And essentially, they do this arousal because they need to sleep. So there is a sleep depth they need to do. So they wake up for a while, they dream, they sleep, let's say, and then they go back in hibernation. But there is also another reason. Do you remember we said that during hibernation, all the immune cells, all the platelets, they disappear from the blood. And arousal, you have the immune cells that now can circulate again and search for infection, platelet, they move again, and then if there are like small wound and so on, they can repair this wound. So it's necessary for the animal to have these different arousals. Just for clarity, because you perhaps will see the two terms. Hibernation is like the continuous process. Torpor is like 1, 2, 3. So 1 torpor, 2 torpor, 3 torpor, hibernation. So sometimes I speak about synthetic hibernation, sometimes I speak about synthetic torpor, but I'm speaking about the drop in temperature and then the increase again. Something sophisticated like this we are not able to do at the moment, but we are going through and we will see. And then what happened during hibernation? So why it's important for space? Because why hibernate? Survive temperature, food, predator, and so on, but then it can be interesting also for space. Reduced metabolism, no consciousness, preservation of muscle strength, preservation of bone mineralization, and then up there we have enhanced radio protection. And we saw that in space we have a big problem with radiation. So enhanced radio protection is what we were looking for. And then there are a lot of studies of natural hibernators irradiated during their inactive state or active state. Most of the experiments were done before the Apollo mission, so are very old. Most of the experiments were done with a very high dose, but you see that there is a clear increased radio resistance of this animal when in when in hibernation. And then also, they find out in this previous experiment that if you irradiate an animal an obligate hibernator in winter, even if it's active, the radio resistance increases. So there is something in the blood of the animal that make the animal more radio resistant. So even if it's not in hibernation, this process that regularly happens every year, it increases the radio protection. Moreover, additional increase in survival may have occurred due to post-time irradiation. So if you irradiate an animal in hibernation and you keep the animal for 1 hour, you have a certain survival. If you keep the animal for 1 day, you have another survival. If you keep the animal for 1 month, we don't know. But in principle, the idea is that you increase the radio resistance. And then now, actually, there are also companies, space agencies like NASA and ESA that actually are studying this. In ESA, there is a topical team dedicated to the hibernation and torpor. So the HIBE project. And also NASA is already, the NASA is already thinking about some concept of spacecraft that could be used for astronauts. Because essentially, if you could hibernate the astronaut, you could reduce the stress of the astronaut to live in a small space for a very long time. You can reduce the amount of food on board, the amount of water on board. You can reduce the spacecraft itself. You can reduce the energy. You have no problem, or let's say you have less problem with the bones and muscles, and you also increase the radio resistance. And now the question is, so is human hibernation possible? Because we don't want to send a squirrel to the moon and Mars. We don't want to send bear to the moon and Mars. So is human hibernation possible? If you observe the order of mammals, you see that all of them have some animals that in principle is able to have some kind of hibernation. Hibernation, daily torpor in green, so only 1 day of torpor, starvation torpor, estivation. So there is always, and you see, all of them, they come from one common progenitor. So they all come from this common progenitor. So perhaps, even in human, there is the molecular mechanism that give the possibility to induce the hibernation, but we just forget about the hibernation itself. For example, many years ago, scientists were doing experiment with mice because try to produce like a synthetic hibernation in mice. And then they find out that actually mice are able to go in hibernation, but they are able to go in starvation, starvation torpor. So they are facultative hibernators. And now you can see this video that is done by Matteo Cerri, that is one of our colleagues that collaborate with us. Look at the mice on the top. So you have this bar. This bar is about temperature, body temperature. No? It's a thermocamera. You see the animal is becoming colder and cold. This is a mice. Just decreasing the temperature of the cage and reducing the amount of food. So you reduce the amount of food, reduce the temperature of the cage, and then you see the animal. It's not going to die. Yeah? After, when you give food and when you rise up the temperature in the cage, the animal will become active again. But you have to trust me, I don't have the video for that. But you see, it's becoming colder and colder and colder to very low temperature. So mice, in principle, they can go in a kind of hibernation. And actually, there are cases also of human that medical doctor could not explain. There are papers on on the Lancet. So these are a peer-reviewed journal. These are scientific journals. These are not like journals that they just give like a random news and so on. These are peer-reviewed journals. And actually, there are cases of human that medical doctor cannot explain their survival if not with something that is similar to hibernation. Anna-Elisabeth Begenholm, she went in a skiing accident. So she was trapped under the ice in a lake. And she was I'm reading because I also do not, 40 minutes in the water. She had the lowest body temperature, 13.7 degrees body temperature when they found her, 13.7. She was able to breathe because she found like an air pocket under the ice, but she suffered circulatory arrest. And that is her in an interview immediately after. So she had like no damage. Erika Nordby, she was 13 months old girl when she left the house unattended, only with a diaper. And then she was in a minus 24 degree. When they found her, no heartbeat, no respiration, body temperature was around 16 degrees. And she's okay. And him, Mitsutako Chikoshi, he was for 24 days on the mountain with a body temperature, at least when they found him, body temperature of 22 degrees. And he also recovered completely. Moreover, if we look at the fetal life of bird and mammals, they are at risk of hypoxia during their process. And then sometime the fetal use some mechanism that is similar to hibernation to do not produce a damage to the baby. So yes, probably, so we are not saying that this is something that it's sure, but probably we can be open at least that probably human hibernation, it could work, probably. And then there are experiments about hypometabolism and suspended animation. So a way to induce hibernation or hypometabolism or suspended animation, we can call with different name in animals. And now this is the case of the zebrafish. It is this nice fish on the upper right part. So essentially for fish, they are able to go in a kind of hibernation, but if you decrease oxygen and temperature, if you decrease just temperature, they continue to have the metabolism working, but they become inactive. If you decrease also the oxygen, creating a kind of hypoxia ambient, they are in a suspended animation and they decrease their own metabolism. And then you see, irradiating this animal, you can see the structure, the muscles of this animal control suspended animation state before irradiation. With irradiation, you are destroying the tissue of the animal. You are producing skin rough, incidence of skin rough on the top. You are even stopping after 74 hours the development of the embryo, while this does not happen when you have the animal in suspended animation. And then with mice, we saw now that in principle, if you decrease the food, if you decrease the temperature, they are able to enter alone in hibernation. But you can also do in another way. So in this case, they irradiate the animal, and then after 60 minutes, they bring the animal in hibernation, but they did with an injection of AMP. AMP is the adenosine monophosphate. Usually, let's say when in Christmas we eat a lot, we try to keep a lot of energy producing a molecule that is called ATP, adenosine triphosphate. And then every time that we cut one phosphate, we produce energy. The AMP is like the lowest energy, let's say, reserve that we can have. So if you give AMP to the animal feel like there is no food, and then it can enter in a hypometabolic state in this case. And then if you irradiate the animal before, and then you bring them this hypometabolic state, you see the blue line are the animals that went in hypometabolic state. And this is Kaplan-Meier curve. So you see every step is like a percentage of animal that is dying. So here you have like 20% of the animals still alive. But for the blue line, after 30 days, you have almost 80% of the animals that are still alive. And then we also start to study synthetic hibernation, but we did it with another animal, rat. And rat, actually, if you reduce the temperature, if you reduce the food, rat cannot go in hibernation. So this is really a non-hibernated animals. The first time that we did the experiment, we did it in a quite invasive way. And this, I have to say that this is absolutely a project that is in collaboration with the University of Bologna. So without Matteo Cerri, that is the expert of hibernation, you can find a lot of video of Matteo Cerri online. So without him, this would have been not possible. Essentially, in this case, on the rat, we went with the cannula through the brain inside a small part of the brain that is called the raphe pallidus area. The raphe pallidus area is important for sleep regulation, body temperature control, mood regulation, and stress response. Injecting GABA agonist, so the GABA is a kind of communication between the cells. Injecting the GABA agonist, we switch off this particular region, and then the animal went in the, sorry, it's not this the graph, went in the lower body temperature, and I have here the picture. So you see the rat before in the cage, before, during, when the process starts and during synthetic hibernation. So here we drop this red line just to show where the animal is, but you see it becomes really invisible in the cage. And then we irradiate the animal with X-ray first, and then in a second moment with particles, because we want to simulate what happened in space. First time was with this cannula into the brain of the animal. The second time we try to inject the 5-MMP inside the rat. We wanted to see if something happened in the rat. And actually, even in the rat, we had a drop of temperature because of the AMP. So this works. So what is the idea? Why we want to irradiate the rats in synthetic hibernation? The idea is because we want to use synthetic hibernation for interplanetary deep space human mission, but also, and we will see in the end, for medical reason. And now I show you just a picture about the experiment that we did it with the X-rays. So this is the liver of the animal in normothermia. This is the liver of the animal in synthetic torpor, irradiated. You can see here there is a vacuolization. There are cells that are dying here, while in synthetic torpor you have like a preserved structure. This happened also in the testis. Look here, this is even more clear. This is the testis of animal irradiated in normothermia. So this will be the structure. So you see here, so there is like a completely disruption of the structure, while here is in synthetic torpor. And then we move to the, of course, we published this work, so again in a peer-reviewed journal, and then we moved to the ion. We wanted to see what happened with the ion. But first we did some tests in vitro. And then the idea in vitro, this is not our work. I just take the GIF online, just to show you the way the cells go in apoptosis. So they are attached to the surface, then at some point they become like globular and they start to form all these corpse like this. So the cells here decide to die, yeah? So it's going in apoptosis. And then here you see this white spot are damage to the DNA, as we saw in the beginning. And then you see, when you decrease the temperature and use the drug, you have almost no DNA damage in the beginning, but the DNA damage appears later. So the idea, we don't know, but the idea is that you have a problem with your car and you want someone from the workshop that repair your car. And then you can travel, you can drive 100 kilometers per hour, and this person can try to repair the car while you are traveling 100 kilometers per hour, or you can go slower. And then this person can do like a better work. So perhaps this is one of the reasons why it happened, but again, we don't know that. However, when we look at the tissues, again, after using particles, now you have the brain. So in the brain, when you irradiate the animal, so when I write saline, it's just because when you do an experiment, you inject a drug, in the control animal, you have to inject just the solution in which the drug is dissolved. So that's why we have the saline. And then essentially you see that saline alone, 5-MP alone, in this case, we are checking for the ratio of one molecule. This molecule is activated on the brain cells, microglia cells, so some cells that are in the brain, is activated when these cells are activated. And these cells are activated when there is an infection, when there is a problem, something to repair. So saline plus 2-gray, you have an increase of this, and you can see also that the cells from this dendritic star shape, they become globular. It means they become active. So they become active because there is a damage that you don't see in the animal irradiated with the 5-AMP. Also in this case, we can see apoptosis, we can see the vacuolization. Here, saline plus 2 gray, you have an increase of hepatocyte apoptosis. In the lungs, after injection with saline, you have a lot of deposition of material. This is material that later on will form fibrosis into the lung. And fibrosis into the lung, of course, is not a good sign. So it means that radiation produced a damage. So we had different publications on this, and I hope that until now I try to, at least, I convince you a little bit more. Perhaps synthetic hibernation is something that we need, at least, to study. It's not science fiction, it could be useful. But why we want to use for medical application? Imagine that we find the right drug or the right procedure to bring a person in synthetic hibernation in a second or in a minute and so on. You will save a lot of life of people with ischemic and heart attack injury, because during this time, all the tissues start to die because of hypoxia and so on. You imagine you have this person, you have like a syringe, a drug, whatever, you put this person in synthetic torpor, and then you save this person. You can preserve the organ that at the moment are bringing very low temperature and so on. You can have modulation of synaptic plasticity, because actually during hibernation, but this happened in the squirrel, you have the brain of the squirrel is amazing, but the brain of the squirrel became like a brain of a person with the Alzheimer. All the cells from the connection that they have, they separate all the connection. They produce a lot of protein inside the tissue, and they become really like an Alzheimer brain. So perhaps studying the synthetic hibernation can help also in this kind of illness. Prevention of muscle and bone atrophy. There are persons that need to stay on bed because they have some other illness, but then they also have problem with muscle and bone. In this case, you can save them. Then radioprotection, chemoprotection, modulation of the inflammatory response, and so on. But also, our hope is that human hibernation could halt the spread of cancer, stopping the metastasis, could stop the tumor growth, and could give us the possibility to treat a patient with a tumor. So now let's consider this scenario. You have a person with, most of the problem when a person has a cancer is not the cancer itself, are the metastases. So from the primary tumor, you have a few cells that detach, circulating tumor cells. They are moving in the blood vessels or in the lymphatic vessel. They go to a distal side, they extravasate, they are quiescent for years. Also in this case, nobody knows why they are quiescent for years. At some point they wake up, they form a micrometastasis, and then they form, micrometastasis and then a macrometastasis. Why this happened, nobody knows. But imagine the hibernation. We are able to stop the release of these circulating tumor cells from the primary tumor. Imagine that we are able to stop the tumor. And now imagine that instead this patient has already like primary tumor and many metastases. Nowadays, these patients cannot be treated. Because if you do surgery, it means that you should open these patients and do too many surgeries, not possible. You cannot do radiotherapy or particle therapy. That is actually something that now exists. So we use the same particle in space to treat cancer. This exists, and we are treating patients with this. But cannot even be treated with this, because the bath of dose that these patients will get, irradiating all the metastases and the primary tumor, will be too high. And then, of course, in the end, the negative effect will be too high. But imagine that we stop the tumor, we stop the metastasis, we increase the resistance of the healthy tissue. Now we can irradiate the primary tumor, all the metastasis, wait that the patient is cured, and then we wake up the patients. And now imagine, we do not have the perfect drug right now. Imagine that, for example, someone say there is a way, but we have to go with the cannula through the brain and so on. There is nothing to do. I guess that even something like this would be an option. It's still worth to study this. And then now we come to some study, because actually now the people are starting to study, many research start to study this. This is an experiment published on the scientific report. They took a carp, the common carp. So the carp, again, the fish. If you reduce the oxygen, if you reduce the temperature, it goes in hibernation. And then here they take a triple negative breast cancer, so very aggressive, and they inject in a mice. So the mouse developed the tumor. This is first in vitro. I will show you the in vivo later, but this is in vitro. Control non-hibernating plasma. They take the plasma from the carp and they put in vitro with the cells, with the cancer cells, and then hibernating plasma. Again, we start to have apoptosis. So the tumor cells are dying because of the hibernating plasma from the carp. Is not happening with the not hibernating plasma, yeah? Here is happening. And then what happened in vivo? They took this plasma from hibernating carpa and then they inject in mice. And now you see this. This is the tumor volume in cubic millimeter on the y-axis. So with time, the tumor is becoming bigger. But then you see control non-hibernating plasma, hibernating plasma, you are stopping somehow the tumor growth. You can see also here, also the tumor weight. The tumor of the animal injected with the hibernated plasma, let's say, they have like a small tumor. And what about the metastasis? They check at the lung and the liver metastasis, and there is a degree of metastasis. So really, the hibernated plasma is reducing also the population of circulating tumor cells that goes to form the tumor. And even the survival, again, this Kaplan-Meier curve, survival percentage, 100% of the animal alive, 0% of the animal alive. These are the animal with the hibernated plasma up to 60 days. 60 days, they are all dead. But up to this point, you have still high percentage of the animal that are surviving. And actually, some time ago, we also start to study now synthetic hibernation with cancer. So actually, this month, we will first time the experiment with animal. Actually, we are using another model of cells. So we're using murine osteosarcoma in mice. So also this murine osteosarcoma, it grows inside the tumor, start to spread, form a lot of metastases. What we want to do is, we want to inject the tumor. Most of the metastases are in the lung. Then we want to irradiate the lung and the tumor to see if we are able to cure the animal and to save the animal, let's say, from the tumors, saving the healthy tissue around. But we started with the first protocol just injecting AMP in the animal. And you can see here, we did it what we try to call like hibernation and not torpor anymore. So we did injection every 2 days. And every 2 days, you see the red are the animal injected with the saline, the black are the animal injected with the AMP. Look at the body temperature. It goes down very much, okay? 22, 21 degrees. This, I mean, we spoke with people that work with hibernating animals, and they say this is hibernation. I mean, this is really because the temperature is too low. And you can see also here, these are the animal now in the cage, in hibernation, and the active animal. Yeah, so we put here some separation, and then we see the active animal and the animal in hibernation. What you see on the upright instead is the rectal temperature of the animal that are not injected with 5-AMP. And this actually is the first result that we get now. Look at this. We are speaking about 12 animals. So it's still like not a huge number of animals, but it's something. So the tumor is growing in both the group of the animal. Then we start torpor. And then you see at some point tumor. It's really like in the hibernated animals, we were able to stop the tumor grow, while in the other animal, the tumor is continuing to grow. What we are going to do now, as I said, is we want to irradiate also the tumor and check if we are able to control the tumor and to spare the healthy tissue. So in conclusion, sorry, it was very long. I hope nobody is in hibernation right now. So I hope I convince you that hibernation has potential for the space mission. Again, not now, not in 1 year, not in 10 years, but there is a potential in many years. Hibernation could help reduce space mission cost. It could minimize the microgravity problems. Most importantly, it can protect from the galactic cosmic rays and from the space radiation in general. Synthetic hibernation, preliminary study. Here I wrote our preliminary study because actually it was before that I saw this paper that actually from these other colleagues. Our preliminary studies suggest that synthetic torpor could potentially be used to treat cancer. Potentially. Again, we are just investigating possibility. What are the future directions? We need new protocol. AMP is in any case toxic. It's not the best. The GABA agonist into the brain, I would prefer not, let's say, also for human and so on. So we need new drugs and we need a way to have prolonged synthetic hibernation. That's important because we have always this small torpor. We need prolonged synthetic hibernation. The good news is that a lot of groups are working on this and there are already nice results. This is the last slide, I promise. On the top, you have mice with an ultrasound probe on the head of the animal. Activating this probe, they are able to switch off a particular region of the brain of the mouse that actually is the preoptic area. And then every time they deactivate the ultrasound, the animal enters in torpor, as you see from the data there. The nice thing is that they find out that there is one molecule that is ultrasound sensitive, and they find out that this molecule is also on the rat. That actually are non-hibernator animals. So perhaps the next step will be to use this probe on the animal. And then there are also other groups that now are studying new drugs, like the donepezil. This is a drug that usually is used for Alzheimer's patients. And they start to use, it's also kind of toxic, but the idea is to encapsulate the drug in nano emulsion with some lipid drop. It's a little bit, not really like this, but a little bit like the mRNA vaccine, where essentially you have the RNA inside the lipid drop that is injected into the muscle. In this case, the drop is given to the xenopus, that is a frog. So this is the larvae. And also in this case, they are able to reduce the oxygen consumption of the animal, reducing the mitochondria activity. So the mitochondria are the fabric, let's say, for the energy inside the cells. They are able to reduce the oxygen consumption, swimming motion, cardiac frequency. So there are other people working on that, and perhaps we can come with some interesting drug or device in future. So the message is, thank you very much for your time. Thank you for the attention. I really hope that at least I convince a bit that this is not a far, far away dream. I have to thank all the people that are cooperating to this work. Again, the group of Bologna is very important for us. People in my group are really the person working in the lab, so these are the most important person. And I leave you with this picture from the Voyager 2, where actually here, inside this blue circle, you can see the Earth. And actually, we are here, our ancestors were here, everybody you know and so on are in this small dot, suspended in a sunbeam. Thank you very much. I think I was a little bit long, sorry.
I think it's also unfrozen. Thank you very much, Walter. It was a science, not science fiction. So
on the edge.
On the edge. But we are eagerly waiting for those things to come to reality. And meanwhile, we start to ask questions.
Molte grazie, Professor, for la sua presentazione. Thank you very much for your good presentation. It was very interesting. It looks to me like the CAMP is kind of a molecular motivator of inducing hibernation. How many
AMP, you mean?
AMP? Yes. See, how many molecular sensors are there for this hibernation process? Is there GABA B included in this?
So
is there glutamate?
the GABA is.
Yeah. And the second question is: did you had the chance to look in species where hibernation is a process, whether they have some similarity in the receptors behind?
So for the GABA, so GABA agonist is something that switch off a particular, switch off the neural activity. So this is something that in principle you can use to switch off the different region of the brain. And there are animals that for entering hibernation that switch off, of course, particular region of the brain. The brain activity still stays in any case, even if they go, for example, the Arctic squirrel, even if they go to 0 degree, they have like a small part that is still active. And this small part actually is able, when the animal has to go out from the hibernation, to reactivate everything, and then they wake up again. For the AMP instead, this is something, this is not really related to the natural hibernation. I mean, the idea originally was to use the AMP because it's like, it's like a molecule that gives, let's say, to the cell the signal that there is no energy anymore. And then the idea was, let's see what happened. And actually, the first animal where they tested the AMP, when they tested the AMP, they didn't even know that the animal was able to go in torpor in case of starvation. Now we try also with rat, and also with rat is working, but with rat is working less than with mice. And in any case, it's toxic. So this is not the solution. The second question, I don't remember, sorry. What was the second question?
Whether these species which are known for hibernation, whether they have some similarity in the molecular receptor structures underlying?
In principle, no. There is like a divergent process. That's interesting. So essentially, all the animals try to use the same mechanism. For example, now there are a lot of universities that are studying the blood of the animal during hibernation to find out if there are inside. And they found what are called like hibernation molecules, they call them. It's not clear exactly what it is. Actually, today, I was searching for a company that could sell these molecules because I would like to test also for the animal and so on. But actually, all the message were, it's like still very private. We are doing tests and so on. So it's like really not communicating. But there are a lot of universities that try to investigate. In any case, it looks like some of these molecules are almost similar to the opioids. So that's also why it's very difficult to sell. But they are studying, they are investigating this molecule, and for sure they will come out at some point. About the AMP, by the way, I still want to add something perhaps. We also try to understand if there is a difference between AMP on the tumor cells and healthy cells. And actually, the healthy cells, they have more receptor for AMP compared to the tumor. So perhaps there is even a better response in this sense. But I mean, this we are still investigating, really the molecular point of view.
Thank you very much. Absolutely interesting also because it is hope, for all the people that are sick. My question is, you are already using nowadays, especially for athletes, hypoxia and also the cold chamber in order to increase, in fact, their, let's say, activity, so that their athletic activity.
Sorry,
how does it? Yeah, in order to get better in what they are doing. Yeah, they train for athletes. Okay, yeah. So how is it possible that you have the same two kind of things, so lack of oxygen and low temperatures, that function for the athletes in increasing their abilities, and with the torpor and the hibernation in decreasing every activity?
For the athletes, they use like low temp to cure the micro damage they have. So they repair their muscles, actually decreasing the temperature. Actually, there is also a nice story about this. During the French Revolution, I think, there were soldiers that were wounded on the battlefield. And then when let's say the emergency come to take this soldier and bring, let's say, they find out that the normal soldier, they had like better recovery, while the officers and all the high-grade military, they had like not better recovery. And they were asking themselves, why this happened? And then the answer is that the poor soldier, they were treated very bad and they were like in a very cold room. While the officers, they were like in a very nice room, perhaps with a camine and so on. And then they had even less recovery. So actually, what the athletes do now, they use like cold temperature and so on to repair the damage. So it's not that they are more active. Before doing, let's say, the match or the run and so on, they have to warm up. But to recover their muscles and so on, they use the cold temperature because this helps a lot. And still the other question, sorry, I have like very short memory.
So
and the hypoxia, for the hypoxia instead is a different story. Decreasing the oxygen concentration, you reduce also perhaps the free radical production. So for example, in particle therapy, so again, do you remember I said at some point that we are using the same particles that are in space also to treat cancer? We use this particle, we shoot the particle into the tumor because essentially they are able to kill the tumor cells when they go slow. And when they are very fast, they have less interaction with the healthy tissue and they produce less damage. However, into the tumor, you have region of hypoxia. This region of hypoxia, they're very radioresistant. Why this happen? This happen because essentially the damage that is produced essentially is like a direct damage to the DNA, but you have also an indirect damage that is produced by the free radicals that in the end produce like an additional damage to the DNA to the cells. If you decrease the oxygen concentration, you have just the direct effect, but not the indirect effect. And you have like a radioprotective effect that actually is called, in radiotherapy, is really called oxygen enhancement ratio. So you have like, if you want to kill a cancer cells hypoxic respect to the normal oxygenated cells, you need like 3 times higher dose or something like that. So in this case, I mean, the motivation can be connected to the radicals production.
I have a very non-expert question, but low temperature and low oxygen reminds me very much of the condition of the alpine explorer that nowadays are not so unpopular anymore. So you should have a lot of statistics in this kind of population. Is it something that is studied? Is it used as a sample to increase?
There are studies on this, but honestly, much experience on this, so I cannot answer on this sense. But yeah, definitely, I mean, there are studies on this, definitely, yeah. But I do not have experience on that.